Okay, what we're doing here today is another question that came to me yesterday from one of your colleagues. And the question reads, if you have a heterozygous parent and a homozygous parent, wouldn't you just foil for both parents and then cross them? Well, that's true, but I just want to show you something. It's an interesting question, and it kind of brought back memories of the problem that I sent to you the last time I sent you a problem set from a student. And that's in the original problem set that was sent to me was if you had a homozygous dominant. So here's one parent who's homozygous for both of these traits, whatever they are. Now, the traits can be anything. They could be molecules like hemoglobin, or they could be color. It all depends on the situation. So you're going to have to read each situation carefully. But what is clear is that this is a homozygous for two traits, this A and this B, whatever they are. And if you cross them with a homozygous recessive for these traits, now notice that both traits are not homozygous recessive. This is a cross. This is one parent. This is the first generation. It's a parent. They're true breeding, so they're, tall and golden, or they're short and yellow, or short and green, whatever this case may be for the traits. But if you look at this, the question is not, wouldn't you do foil and then cross them? You would first. The reason you do foil is not to cross them, but because the crossing is happening when you exchange the DNA. The key is what are the gametes after meiosis? What are the possible gametes after meiosis? So we're looking for the gametes, and it's the gametes that are going to come together during fertilization to create the zygote, right? So two gametes, whatever they are, and it could be sperm and egg, it could be pollen. It could be whatever, but the key is the two gametes, two individual cells that have been reduced to an N number, whatever that N number is. So from 2N through meiosis, you've gotten this two. This is the male version, and this is the female version, right? And so they come together, and they're going to join, and they're going to form one zygote that's a 2N, right? So in humans, it was 23 single copies and 23 single copies to give you 46, right, total, or 23 pairs, right? And you remember that from meiosis, right? Okay, so when we're looking at this and we're seeing this homozygous, and this is homozygous, right? Homozygous, and what again? Goes back to the vocabulary that you guys have to work on. So homozygous means that they are the same, right? Homo means the same genes, right? And this is homozygous, so they're both homozygous. But how do we tell the difference between these two? Because obviously they're different, right? If they're both homozygous, how are they different? Well, they're different in that these two are what? These two traits for this parent, and let's say it's the female. This parent is, these two traits are what? I hope you said they're dominant, because up to now that's all we've been talking about is dominant recessive, right? So these two traits are dominant, so we call them homozygous dominant. And the other one we call homozygous recessive. And it's really easy to get. I think it's easy. You have two traits are homozygous. How do I know they're homozygous recessive? Because they're small letters. And how do I know they're homozygous? Because they're the same, both small a, both small b. Well, how many possible gametes can the homozygous dominant make? I don't have to do foil, because this homozygous dominant parent can only give a big A. That's all it has. It can only give a big B. So the only gamete that this male or female homozygous parent can give would be big A and big B. That's all it has. So I don't have to do foil. There's not four possibilities. There's only one. And so in the case of the homozygous recessive parent, the male in this case, there's only one possible gamete it could have, it could give, which is small a, small b. So here, really, the zygote that it's going to make, the only zygote that these two true breeding individuals could give, is a heterozygous for both traits. So that leads us back to the original problem that we discussed in the previous video, which is if you're crossing this homozygous dominant parent with this homozygous recessive parent, the only gametes, the only haploid gametes, right? Because these are haploids. The only haploid gametes it can produce are these are the gametes that you see here. So when we're talking about, what is it that we're producing? We're producing this zygote, and the words, see these vocabulary words just keep coming back. This zygote is the new human, the new plant, the new whatever, that is heterozygous. This is heterozygous. So this heterozygous parent comes from this homozygous dominant and homozygous recessive being crossed, producing their haploid versions of themselves, which there can only be one version because they're homozygous, producing this heterozygous offspring, and this would be your F1 generation. And what you saw in the previous problem that we discussed is that the problem takes these two of these, if it's a pea plant or carrot, that's easy to do, two of these heterozygous individuals from the F1 generation, and it crosses them. So then you have to do FOIL to figure out what are the possible gametes, and this individual can produce.